Good afternoon, morning. Welcome to this week. Today we're embarking on a critical chapter of human biology, which is embryology and development. Our goal is to trace this complex journey from fertilization to early embryogenesis. This knowledge forms the basis of understanding everything from congenital anomalies to advanced reproductive technologies. So let's dive in. Before we begin, we want to get our terminology straight. The term conceptus refers to developing offspring, regardless of stage. Pregnancy describes the state of carrying a conceptus. The gestation period begins from the last menstrual period and continues until birth. And we define the embryo as the conceptus up through week 8, and from week 9 onward, it becomes a fetus. These distinctions are fundamental when correlating developmental stages to clinical practice. Fertilization is the starting line. It's when the chromosomes from the sperm and the egg combine to form a single diploid cell, the zygote. It's highly coordinated. It's a time-sensitive event, and coitus must occur within a tight window, up to 2 days before or within 24 hours after ovulation. Understanding this timing is vital in fertility and assisted reproductive or reproduction settings. Once the sperm meets the egg, the real action begins. Fertilization involves the sperm's genetic material merging with the oocyte, forming the zygote. But for this to happen, sperm must be present and viable in the female reproductive tract when the oocyte is released. Now, the tight survival windows are important to note. The oocyte lives for 12 to 24 hours, sperm up to about 48 hours after ejaculation. And this is why timing is everything in conception. Not all sperm, though, are created or even prepared equally. Sperm undergo a process called capacitation in the female tract, or the AFAB tract. This is a crucial step that makes them competent to fertilize. They gain enhanced motility, and their membranes become fragile enough to release enzymes. These are enzymes that are necessary for oocyte penetration. Fascinately, they also use olfactory-like receptors to quote-unquote smell their way to the egg, which is chemotaxis, essentially another example of chemotaxis, like we see with our immune system. Despite the millions of sperm that are released during ejaculation, only a few thousand actually make it to the uterine tubes. Many are lost or destroyed on their way there. This could be due to vaginal acidity, barriers from the cervix, or even immune defenses. This attrition emphasizes the improbability and biological precision of fertilization. It's a numbers game, but only a select few will make the final cut. The next step after this process is the acrosomal reaction. This is triggered by calcium influx, as so often many of our cellular processes are. As sperm reach the zona pellucida of the oocyte, they release hydrolytic enzymes, things like hyaluronidase and acrosin. These enzymes work together to digest through the protective barrier, setting the stage to allow for membrane fusion. This is a cooperative effort, and many sperm will help clear the path for one who finally will reach the oocyte. In the approach, the sperm uses surface enzymes to weave through the corona radiata, which is the outer layer of follicular cells, as seen in this picture. As they bind to the receptors in the zona pellucida, calcium channels will start to open in the sperm, which initiates the acrosomal reaction. This cascade allows sperm to bore through the oocyte membrane. The specificity here ensures that only human sperm can fertilize a human egg, reproductive compatibility that's encoded at the molecular level. And we can see this in this picture here. This is showing the approach, which is aided by enzymes to work through this superficial layer. The chromosomal reaction will intensify here as multiple sperm continue to contribute enzymes to break down the zona pellucida. This redundancy ensures success. Without the cooperative enzymatic assault, the oocyte's tough glycoprotein coat would remain impermeable. It's another reminder that while fertilization is a one-on-one event, it's enabled by a team effort across the board. Finally, the last stage is binding. One sperm will bind to the oocyte membrane, which will trigger the oocyte to form microvilli that wrap around the sperm head and fuse the membranes. This step will ensure that only one sperm nucleus enters, initiating the final, irreversible steps of fertilization. It is truly an elegant gatekeeping mechanism because once this binding occurs, other sperm are blocked from entry. At this point, the membranes of the sperm and the oocyte will fuse together. The sperm's cytoplasmic contents, which includes the nucleus, will enter into the oocyte. However, the tail of the sperm and the mitochondria of the sperm are left outside. This detail will have clinical implications. Since mitochondria are maternally inherited, disorders of mitochondrial DNA are transmitted exclusively through the mother. This fusion will initiate the final phase of fertilization. Now, I mentioned it's a one sperm per oocyte problem typically. And so once one sperm has successfully entered, the oocyte must prevent others from doing the same. There are two mechanisms at play, as we can see here. First, the oocyte membrane block, where the oocyte sheds all remaining sperm binding receptors. And second, the zona reaction, which is a cortical reaction in which enzymes will harden the zona pellucida, which renders it impenetrable. These checks will ensure that only one sperm's DNA will contribute to the zygote. We'll stop here. When we come back, we'll talk about meiosis II and the process of fertilization continued.